Let me see. I picked the test up today. Let's see how big this stuff is. Thank you.
I didn't come off those ones, I forgot. I don't know. Oh, I was trying to get out of bed.
Is there actually oh, there is one? Did you know I did it?
I was like trying to crawl out of bed at 7:30. That hurts.
80 right here. Now you know that there's no way I use all these questions on this paper test. Because there ain't that many. But right there's 80. If you figure 80, and that's 300 something answers. I ain't got to take a test. Problem is though, I don't recall which ones I used. I don't recall. But I'll share with you.
Okay.
Lesson 5. My thought is, why make up new questions? I got them all.
I like that.
I get them right here. Now, once you answer that first one, carbon. And carbon is the thing that everything that's organic is built around carbon. And what's right here?
Could you say, would you be able to say hydrogen for the first one too? Because there's carbon and hydrogen? Or?
No, you got to say, if you had to pick one, you better pick carbon. Now, if you're saturated, then how are you at room temperature?
Solid.
If you're packed full, you're a solid. There's no room to be a liquid. If you're unsaturated, you're not packed full, right? Then you'll float, and you're a liquid. Now, this next one here, which of those combinations would truly be an organic combination? Okay, alpha would be and Bravo would be. The rest of them, I see my C's, but I got to have a C and a C or a C and an H. That's only my first two. On this one, how many bonds can carbon form?
4.
4. That's why it's such a good thing. It's got 4 arms. They can grab 4 things. It makes a great bonding agent. 2 of those make sucrose. What 2 are they? Glucose and fructose. I wouldn't care about the order you put them in, because my key has them. You can say B and C, or you can say C and B, and I won't mark you wrong either way. What are 4, we tested for, what are 4 main organics?
Carbohydrates.
Carbs,
Nucleic acids.
proteins, lipids, and nucleic acids. Of those, the most complex type, 2 of them are the same thing. Which 2 are the most complex? The starch and the polysaccharide. A starch is a polysaccharide. Most complex thing there is. B and E, by the way, are the same thing. C would be just 2 monos put together. But poly would be, so many you can't count them. So I have starches that way. Carbohydrate, what's it really mean? You only have what kind of atom? What do you think carb stands for? Carbo is almost what word?
Carbon.
Carbon. Hydrate stands for?
Carbohydrate.
No, hydrate. If you become dehydrated,
So you dehydrate it?
it's water.
So carbo water?
Only
Carbon water?
read it. Some kind of atoms and some kind of molecule. I didn't say atoms both times, did I? I said, now, if I say what kind of atoms, that answer is what?
Carbon.
And then when I said molecules,
Water.
now I'm going to reverse that sucker and say composed of blank molecules and blank atoms. If you memorize the blanks, here's what happened to you. You're going to get it exactly wrong. Learn the stuff, because I'm bad about swapping things around to get folks to memorize in trouble. So when I say atoms, you say carbon. When I say molecules, you say water. But I might reverse it and have molecules come first in my question. And you know I do that kind of stuff. Only 2 of those are carbohydrates. Because here we look for it. They've all got carbon, right? Now what's the formula for water? It's always 2 to 1, right? Now find the 2 up there, which is 2 to 1 water.
See.
Okay, see A up there? 4 to 2. Ain't that 2 to 1? That's fine. And look at number C. 22 to 11. Ain't that 2 to 1? Now look at 21 to 7. That's 3 to 1. Is that water? 46 to 20. 77. Are those 2 to 1s? No, that's how you do it. You look for the ones that are 2 to 1, because those are the waters. And by the way, the others on there would be fat. If you go bigger than 2 to 1, you're fat. Same 3 elements. Same 3 elements. It's just that because you've lost the water, you're now fat.
So it's A and C?
I don't know what they are now. Don't say A and C.
Well, it's
Because it won't be A and C ever again. I'll guarantee you that. It might be Z, Q, R. I'm going to mix them all up. You need to understand that the last 2 letters have got to be water. That's going to be a 2 to 1 ratio all the time. And it happens this time A and C. But don't count on that again.
You mean A and B?
A and B, I'm sorry. No, it's A and C. It's A and C. B is not 2 to 1. Look at 21 to 7. That's 3 to 1.
Oh.
21 to 7 is a 3 to 1 ratio, right? That ain't water. Can't use B.
Okay.
All right. Which 2 of those make maltose? You always say what?
Glucose and glucose.
You always say glucose, but this time you do what? You gotta say it again.
Yeah.
Maltose is 2 glucoses. You gotta say C. You gotta say B, and then B again. But don't do it tomorrow though, because it won't be B. I use the wacky, weird letters, A and T and P and Q and R. Well, now, what kind of structure is that? This time I didn't write down.
D.
Huh? That's the quad.
That's what?
I got 2 of the yellow worms. I got 2 of the blue worms. 2 and 2 equal what number?
4.
Quad. Quadrant. Dehydration, in other words, if you dehydrate, do you add or lose water?
Lose.
If you synthesize, do you join or take apart?
Join.
Which of those shows losing water to join?
What is added to join? No, what is removed to join? B.
First off, you gotta look for remove, right? So
Yeah.
it's gonna be B or D, right?
B.
Then you figure, are you joining or separating? That leads you right back to Bravo. Because you remove the water, that frees up a bond forms, and then they glue together. Organic molecules, they never have, always have, sometimes have, what's the answer? They never have that metal in them. Now, what you see there is a disaccharide losing water and becoming, oh, I'm putting water back.
So separate.
This says, I'm putting water back and making this happen. I'm putting water back, and the water goes back. You see the O right there? But you see that the 2 H's and the O right there? That's the water put back. And now they separate.
So they're breaking up so that
So they're breaking up.
they
So first off, ana or cata.
cata,
Second off, hydrolysis or dehydrolysis.
hydrolysis.
Hydrolysis. You're not making anything. You're not making anything. Now, if you have a question, speak out. Now, I'm not looking at hands. I'm just talk out loud.
This anabolism.
All right? Where do you find all the energies in sugar?
And the bonds.
And the bonds themselves. When you break the bonds, you get to enjoy the energy. Stiff starch holds a plant up.
Cellulose.
And can you or can you not get anything out of it?
You can't.
You can't digest it. It forms a roughage in your diet. You got to have it, by the way. When you eat lettuce and black-eyed peas and broccoli and squash, you got to have the cellulose to make your intestines work. But you can't digest it and make food out of it. Saturated fats, where you get them from?
From animals. Saturated fat.
Which of those is not a great big old polymer? the amino acids. Proteins are made of amino acids. Starches are made of monosaccharides. Nucleic acids are made of nucleotides. If you're made of something else, then you're not. Then you're a polymer. Amino acids, they make proteins. They're the ones that are not the polymer. They make the polymer. Which of those two are the most common ways your body uses carbs? How do you use carbs?
Energy.
Supplying energy.
You make the energy with carbs, and what else?
Creating structure.
Create structures.
Carbohydrate.
Those ID tags on cells,
Supplying energy.
that's carbohydrate. Hormones are made by proteins and lipids. Enzymes are made of proteins. But which is that one? What is that kind of structure? It looks like a letter E to me. That's the lipid. That's the lipid. Looks like an E. Oh, by the way, saturated or unsaturated?
Saturated.
Yeah, got all H's in the hole, right? There's no H's left over. That sucker's a solid. That's lard. That's lard. Well, what kind of structure is that?
Secondary.
It's starting to wrap back on itself. That's secondary. If it's just one long line, that'd be primary. All right, now we're going to decide sat or unsat for these. Say meat. What's the meat contain?
Saturated.
How about butter?
Saturated.
Olive oil.
Unsat.
That's why it's so good for you. How about avocado?
Unsat.
Canola oil.
Unsat.
Breast milk.
Saturated.
Very saturated. Soybean.
Unsat.
Coconut.
Saturated.
Saturated. Coconuts are bad for you.
That's confusing.
I love them. I should know right there they're bad for me. So I love them. Coconut's the one you don't want to eat if you're diabetic. And I love a Mounds candy bar. I love coconut cake. All right. Which two of those make lactose?
Glucose.
There was glucose and what else?
Galactose.
And galactose. It's always going to be glucose. And then another member. Simplest type would be, there's two answers of course. Simplest type.
Simple sugar.
The simple sugar, which is also called a
Monosaccharide.
monosaccharide. Those are both the same.
What was it?
No, there's two numbers up there. I require two of them. See 45, 44, 45? So you can't be one or the other, it's got to be both. Far as your health, which of those is good for you?
Unsat.
Unsats. Which one's good for you?
HDL.
HDL. Think of happy.
Yeah, that's what.
Unsaturated. What do they do?
Lower LDL, raise HDL.
They lower the bad LDL and they raise the good, so alpha's the answer. Good thing it ain't B.
Yeah, that'd be upsetting.
Unsats.
Plants.
Plants. Except for coconut. That's a weird sucker.
Coconut's the animal.
That's a fruit.
Fruits.
But it's a plant though. Unsaturated. How about them?
The liquid.
That's your oil. That's your,
The
pour out of
liquid.
a bottle. That's your oil.
Liquid.
Steroids are,
Liquid.
that means they can.
The can.
Steroids are lipids. The cell membrane's lipid. They go right through. So lipid and can.
Primary structure.
That's primary. There's nothing simpler than that. It starts wrapping bone stuff and wrapping back on itself. They don't dissolve in water because they are
Hydrophobic.
hydrophobic. You don't like water, you're hydrophobic. If you like water, you're hydrophilic. What's that one?
Tertiary.
That's tertiary. That's one step from being four. I need three more of those things to get four.
Oh, can you wait 1 second? I got a battery change right here. Okay.
You got them?
Yep.
If the only one of those that can change its shape,
Protein.
open the door, close the door, is a protein. Now this time, I am removing water and making them hook together. So first off, they're joining, that means it's going to be
Anabalance.
And I'm removing water to make them join, therefore it's, there you
Synthesis.
go. Enzymes, we end with what? We use ASC. Not always,
Most of them.
but most times. Which of those two is unsaid? Yeah, I got some gaps in those teeth. All right. Now I want to know D or R for these. So if you got ribose, you must be
RNA.
R. Single strand is
R.
R. Helix,
D.
D. Double strand,
D.
Got deoxyribose,
D.
The larger of the two,
R.
R is.
R.
Deoxy, you lost oxygen. So you're a little smaller than the R. But you still got the oxygen. So if you deoxyribose, you take ribose, take off the oxygen, and you have deoxyribose, which is a little smaller. Which one's got uracil?
R.
R. Who's got thymine?
D.
Now, that'd be all shuffled up in my test, so not ordered off. Yes.
Deoxyribose is DNA, right?
What now?
Is deoxyribose DNA?
Oh yeah. Okay. D means deoxy.
So what are the different proteins in the DNA and the RNA? You said there's
Okay.
4 different.
Okay, there's 4 different ones. 5 if you play with U. Okay. The 3 that are in all of them is the A, adenine, C, cytosine, and the G, guanine. Those are in both of them.
So
ACG are in both of them. So if you have DNA, it's ACGT. RNA, ACGU. And the kicker is the U and the T.
okay.
Okay? Polymerase acts like a what? Is it adding things or taking things apart?
Put them together.
Put them
It's
together.
adding things, therefore it's going to be anabolic. They're the choices. I'm going to have to go back and forth to do them all. But let me see, lipid protein transfer, nucleus, ribosome, nucleolus. I got to remember them. All right. Messenger RNA carries instructions from the nucleus out to the cytoplasm. Now in cytoplasm, after reading the messenger RNA, transfer grabs the correct amino acid and carries it to the ribosome. Not ribosomal, ribosome now. So if you're working too fast tomorrow, you might choose H, but E is real answer. So watch out. To the ribosome. The ribosome attaches the amino acids together, producing a protein. A cell's ribosomes are made by the... Who makes ribosomes? The nucleolus does. Now, I don't know how I'll work this on a paper test tomorrow. I may have just a big paragraph and all these choices under
Did
it.
you say nucleus or nucleolus?
For which one?
The last one you just did.
The nucleus does not make the ribosomes. The nucleolus makes the ribosomes. The nucleus contains the DNA, which has the blueprint for the ribosome. The nucleolus makes the ribosome.
But the mRNA.
Who's saturated? Yeah, it's full. There's no room, any more H's at all. What is that thing? It's a ribosome. The small unit separates. The messenger goes inside there and just click, click one at a time. It reads the messenger and brings in the acids, hook them together. The next thing you know, you have a protein winding out of it. True or false? Does every bit of DNA carry something that's important?
It's false.
No, some is junk. Sometimes, like the sun out there can mess your cells up, and all your body does is take that little strand and kind of nullify it and make a new strand next to it. It's all still there, but the one that's damaged is ignored. So not everything in DNA is used to make protein. Not everything. Why do you need enzymes to keep yourself living? without the enzymes, you will not have the chemical processes happen fast enough.
Everything will
You'd
accumulate
eat
in your
food
body.
today and digest it 2 years from now. Well, you'd be dead by then.
Yes.
I need to digest it right now.
So it wouldn't break down the food, you wouldn't break it down into energy? That's what you're
But you want to indicate in a timely fashion. It's going to do it over time. But I don't have a lot of time to wait. I need my energy right now. And enzymes let it happen right now. It would break it down without the enzyme. But you'd be dead before it, because I need the stuff right now, not next week. And that's why the enzymes are so important. But a fever destroys the enzyme in your brain. And that's why the fever kills you. It kills the enzymes that control the processes in the brain, and that's why you die. It does come fast. It doesn't take long for an enzyme to be knocked out. A high fever, like over 113 degrees, is almost always fatal. 108 degrees, if we can keep it right there, we'll pray for you, but you're probably going to survive if we can keep it going higher. But you get 113, I don't know if anybody that survived that high temperature, because it actually destroys the enzymes in the body.
If you survive,
And when the enzyme is destroyed, basically,
brain damage.
I'm killing you.
Denatures all your proteins. Like
It's called denaturing.
If
It's
you
called
survive, you might have some brain damage.
Huh?
If you survive 113, you will have brain damage.
There are a lot of things that can cause the enzymes to denature. One is pH. The wrong pH will make it bend out of shape, so you can't use it anymore. But mainly temperature, heat will make it bend out of shape, and it won't bend back. They might cool you back off. Damage is done, but why are the most effective pesticides lipid soluble?
Because they can pass through
because they
the.
get through the cell membrane.
If you want to kill a bug, but you can't get into the cell, how effective will you be?
No.
It will work.
So if I want to kill the bug, I must get into the cell. So I'm going to make my pesticide lipid soluble. It then goes to the cell membrane. And if I kill the bugs or the germ, then I'll kill the germ. That's how you, when you spray things in your house or working, that odor you smell, that's lipid soluble. But you also worry about your dog licking it.
Yeah.
They'll get him too. They tell you, I mean, these things you put down there, be real careful. All right, let's look at the next test.
Okay.
Oh, yeah.
I'm gonna need more paper.
You need some paper?
No, I have no paper. I pulled out 3 sheets.
Only 35 on this one. Okay. Which of those is the phosphate part of the
A.
phospholipid?
It's phosphate part.
It is the marble, the head. Okay?
Which is the phosphate part? A.
Because that, do lipids dissolve in water?
No.
That's why the tails are far, there's water out here, and there's water on this side. And the tails are as far away as they can get. That guarantees you keep that shape. The tails like this. If the tails of the water, it reverses itself, they'd be sticking out. And the 2 sets of blue marbles would be inside. Alright? Besides those 2 things, what other nutrients must you obtain? What must you have also from the environment, besides oxygen and water?
Carbohydrates.
Carbohydrates,
Minerals.
minerals, what else?
Mineral
Proteins,
Lipid?
lipids,
Wait,
vitamins.
Minerals.
But we already said minerals.
so do vitamins and minerals count the same?
There are a lot of choices you could have picked out for this one.
I have 5.
You know, you buy plant fertilizers called 13, 13, 13, or 8, 8, 8. Do you know what that number stands for?
Nitrogen.
One is nitrogen, other is phosphorus, and another one is potassium. Those 3 chemicals do one of them works on the roots. One of them greens up. If you just put on ammonium nitrate, it's going to make your stuff so green, but doesn't do anything for the root system. It's going to green it up. A lot of golf courses, before they have the Masters tournament, they'll go out there and put only ammonium nitrate down, just all ammonium nitrate, to green the golf course up, but don't help the root structure. During the other part, they put down triple 13 and triple 8. That's going to also get the roots too, so your grass grows. And then lime, you put on lime to control the pH. I had my garden limed about a month ago, because as the plant thrives, it pulls out and it becomes too acid. I need to be basic. So they came out and did a test, and I said, yeah, it cost me $104. But I want a pretty yard, so. And I could afford it, so I did. Which is a channel transport protein?
C.
It lets something go in and go through it and get into a cell. Which two of those is the one that's in the cell membrane? Three of them are not members of the cell membrane.
Sugars and minerals.
What you say?
Sugars and minerals.
Sugars are not in there. Two of those are actual.
You're asking.
We're asking for the ones that are not in.
That are not.
I'm sorry. You're right. Sugar cake. And the sugars are one of them.
Sugars
And minerals.
And minerals. Proteins in there. That's those channel things. Cholesterol is there to hold the thing together. And the phospholipids, you saw the blue things in the tail. So the two sugars and minerals are not there. What is that thing?
The bilayer.
What is this?
I would just said it's a cell membrane structure. The nucleus looks just like that. The nuclear membrane looks just like it. I wouldn't say cell membrane, because if you look at the membrane around a mitochondrion, there's two of those things back to back. One of them represents the mitochondrial membrane, and the other one represents the cell membrane the mitochondria inside of. But that is the basic structure of any membrane, and not just cell membrane. I reckon when the body figured out it like this, that I'll use that thing over and over again. Yeah,
Can we use phospholipid bilayer?
I'll take that.
Okay.
But I'll just say membrane. That's what it is. I mean, you can say that. If you have room to write on my answer sheet.
Plasma membrane would be right.
Yeah. Or nuclear membrane. It is a typical membrane structure. It could be the cell membrane. It could be a membrane surrounding a mitochondrion. That is how the membranes are made. Now, where you look, it looks like that. All right? Three of those words mean the same thing.
Semi-permeable, selectively.
Semi is one of them.
Selectively and differential.
Selectively is another one.
Differential.
And differentially is the other one. Bravo, Delta, and Echo, because they'll be changed tomorrow. Dipermeable, I made those two words up. They're not words, they're just body tap.
Wait, which ones?
A and C I made up.
I thought so.
Now, some folks still picked them, I feel good. Which one's a receptor transmembrane?
Receptor transmembrane?
Trans means to go through. Oh, I see only two going through. You know what one of them is, what's the other one? Echo had to be the one that is the receptor.
Wait, you?
Echo is the one.
I see it again.
Then what's A?
A
Phosphor.
A is a phospholipid. I mean, A is a glycolipid.
Glycolipid.
It's a lipid combined with
Phosphor.
nope. Back up.
Protein.
It's a glycoprotein. Because see the purple thing? There's a protein.
Mm-hmm.
See the blue, all the blue stuff?
Yeah.
Lipids. If you come off the protein, you're glycoprotein. Now look over there at delta. What's it coming off of? Protein or fat?
Fat.
That's your glycolipid. Those are what you come off of, what you are. They're both glyco something, because I see the carbohydrate part. But the bottom part on this side is the protein, and delta is coming off of the fatty substances. Genetically, what do you call those recognition markers that are inside? Yeah, the glycolipids, which I just showed you, and the glycoproteins. That's the name tag. They cell wears so the other cells know he belongs here, he doesn't. If something shows up and is not recognized, it's probably sticking off a bacterium trying to get you sick, and you get it. Problem is though, if I give you one of my kidneys to save your life, will anybody recognize my glycolipids? It's going to think what?
Foreign cell.
And it's going to try to do what to my kidney and you?
Kill it.
What we call that? Organ rejection.
Okay.
So that's the problem we have. And of course, that's why the closer the person is kin to you, the more likely the success is going to be. The glycoprotein, now you should know it's the one off the protein. What's that bad boy?
This is a receptor.
That's the receptor. The round side won't fit in there. Only the pie part, the triangular part will. Well, where's the lipid? There's all those tails sticking out. Whoa, whoa. Yeah, it's all the tails. Look at B, look at C. Which one is inside the cell? That's what intracellular means. Hotel is inside the cell. Which one of those would have to be hydrophilic? Loves water.
A.
The A would be hydrophilic.
Hydrophilic.
What kind of protein is that?
This is transport protein.
A
Transport. Carry something through it.
Yeah.
Nothing else can even be, even look like it. The fluidity in the membrane is crucial to function in those animals. The lower legs get less blood, therefore less heat loss, and they survive the winter because they don't freeze to death.
Caribou.
You ever seen a caribou's leg? You wonder how can it hold that big old body up? They're so scrawny.
Well, okay, it's not that much.
But moose legs are much bigger. Polar bear thrives in the Arctic, as you know. So which one's outside? Bravo is outside. All right. It's got to be fluid. Now, by unsaturated fats, which one's true?
It depends on the
The more unsats you have, the more fluid you'll be, the stronger the cell will be also. The less you have, you're a weakly person. Which one had to be hydrophobic? Hate the water. The ones inside there. Get as far away as they can.
Well, he's about 24.
Cholesterol is also important. Which one is true for cholesterol? Which one makes it more fluid? The more you have, the more rigid it is. So the less you have, the more fluid it is. The answer is bravo. The less you have, because see, cholesterol straps it down. So the more you have, the more strapped down it is, and the more solid it is. The less you have, the less strapped down, and the more fluid it is. Exactly right. What would that one be?
This is a
That's the recognition. That's your name tag.
Recognition.
So other cells can recognize him as being friend or foe. Now, glucose likes the water. It's water soluble. So, is it going to be able to pass through the membrane, or it got to go through a door?
One through the
Got to
door.
go through a door.
B.
That'd be bravo. Because it can't go through the membrane because if you like water, guess what you hate? Fat. What's the membrane made of? Fat. You ain't going going through. Door you can go through. That door is that Bravo. What kind of what kind of proteins are those?
Enzyme.
I'm changing something.
Enzyme.
Enzymes change something. Enzymes change. Is that one cell membrane or two?
Two.
It's just one.
It's one, but
It's just one.
It's one.
And I see what you say two for. You see two lines of those red, round blue marbles. But that's just one membrane. That's just one membrane. Like an ice cream sandwich. Just one sandwich. Steroids are fat soluble. They like the fat. So what's true for them? They can go on through. They don't need that door. Like a ghost. They go through the wall. What would those be?
Adhesion.
There. Now, okay, look at that. How many membranes see right there? One or two? There's your two membranes. Okay, see? There's one membrane, and there's the other membrane. And this is making sure that those two cells stay glued together. But now that would be two membranes. Where's your glycolipid? It's the one coming right off the fat, right? That'd be delta. Now, I got like a connection opening between two cells. That's communicating. They can communicate each other. And nothing else even fits what that could be. That's for communication. What's in one cell can go to the other cell, like send a message across. All right. Which body system depends heavily on those recognition proteins to keep you healthy? The immune does. It makes the antibodies to kill the germs. The immune does. That's all of that one. I often wonder if I'll get on this one. All right. What do you think? Small molecules, speed up or slow down? Speed up.
Semi-permeable, differential.
I will take semi-permeable.
Differential.
Selectively permeable. Differentially permeable. I tend to say semi because I'm lazy. That's easy to write that. Small molecules pass through the cell membrane and go into. Those would be what kind of molecule? If you can pass through the membrane,
Hydrophobic.
you're hydrophobic.
Phobic.
Because you hate the water,
You like
you love the fat, and you go through the fat. Hyper is not hypo, is it? What's the Bobby Ted rule for hypo?
Oh,
The big O.
swells.
Now think backwards. Hyper
Hypertonic
is going to swell or shrink.
To
It's
shrink.
going to shrink. So to shrink, what must happen?
Lose
I
water.
got to lose water. These cannot pass through a membrane.
Hydrophilic.
So they got to be hydrophilic. They love the water, but the membrane not made of water. Membrane made of fat. Hydrophilic and fat don't get along. Which of those cannot go right through the membrane? That big old glucose. It's got to go through a door. The gases can go right through. And of course, steroids, which are lipids, can go right through. What two kind of proteins can move things into a cell?
Carrier protein.
Carrier and
Tunnel.
Channel.
channel. I made tunnel up. I didn't make up D&E, but I did make tunnel up. Fluid, gradient. Okay, that's the choices. Let's see what the slides, if I remember. It's a physical difference in regions make them move back and forth.
Gradient.
That'd be gradient. I'd move from one to another. If the gradient is equal, any movement going to happen? No. All right, next one. It can move and change shape in response to external forces without breaking apart.
Fluid.
The fluid. I know water's a fluid. What else is a fluid?
Gas and
A gas.
and liquid.
No, this ain't a fluid. But a gas is a fluid. I didn't say, you know. So that, a lot of folks don't understand that a liquid ain't always wet. The number of molecules in the area,
Concentration.
it would be concentration. I'll probably take those and make a question and put those 3 choices under. Another question, 3 choices. Another question, 3 choices. 2 kinds of fluids, we just talked about it.
Gases and liquids.
Gases and liquids.
Gases and liquids.
Low temperatures do what to how fast diffusion occurs?
Decrease.
It slows it down, decreases. Higher concentrations make it go faster or slower.
Faster.
It'll increase it, go faster.
Faster.
The physical rules. Molecules are always in constant
Motion.
and they make random collisions with each other. When you boil water, you make the collisions happen more often. And like you hit a pool ball too hard and flying off the table. It happens, the water flies out the pot. Next thing you know, the pot's dry. You keep heat going, you're going to warp your pot. You ever have, you'll be cooking oatmeal and somebody call you and forget oatmeal when they're cooking? You come back later on, it's gone past cornbread, it's almost like a brick.
I did that.
Cook on the water out of it.
It was very exciting.
If there's a gradient, there's more here than there. So molecules move up or down that gradient. They'll go down like downhill. They can go up if I push them, but that's active transport then. Diffusion does not. Happens by itself. I ain't got to make it happen, does not. Passive transport, do you or do you not need the energy? That's without. Moving up or down the gradient? Down. Flows downhill by itself, does it? So is the car. What's that showing you?
Diffusion.
That's diffusion. Spreading out to us equally. The gradient, this is the high part, the low part. And the diffusion goes downhill till it's all full. Now, is there a gradient here?
No.
No, they're equal. Diffusion stops. Is there still a gradient here? Yeah, but not like it was there, though, is it? So the gradient exists. It doesn't exist and things are fine. If you make tea outside the sunlight, you put your tea bag in there and it sits in the sun, right? There's a gradient there and the sun is going to heat the water up, make the molecules move more and more. And very slowly you're going to see your clear water turn a little browner, a little browner, a little browner. There'll come a time though when the gradient is equal, there's no more diffusion and your tea is done. That's why I always like day-old tea. The tea I made for Thanksgiving, Thursday, will taste better on Friday to me. It just tastes, I don't think my mind either. And I would make tea outside, but you guys started Tuesday to make it for Thursday. I don't think it started Tuesday, so I went there and boil the water and put the tea bag in and take out the water and make it fast. But I tell you right now, that the tea I'm drinking now is really good. The tea I drank Thursday is all right, but it's better now. All right? Cell has some or no control over diffusion.
No control.
None at all. It happens by itself. Otherwise, it wouldn't blow up. It wouldn't shrink up. It can't help it. Basically, diffusion rates are pretty
slow.
They're pretty slow, and they cover
a small.
Pretty small. They don't go very fast nor very far. Now.
This messed me up.
2%. Okay.
This messed me
The
up.
bag is 2%. The beaker is 13%. The bag is 2. The beaker is 13. That combination would be hyper or hypotonic. Hyper. If you're hypertonic, what's going to happen to you? You'll shrink. Answer is A. Okay, higher temperatures do what to rate of diffusion?
Increase.
Lower temperatures would.
Decrease.
Yeah. In a hypotonic situation, which of those is true? Two are true, by the way. Two are true.
Would it be the cell contains less than the solution?
A and B.
Okay, first off, the word hypotonic, is it talking about the cell or the solution?
Solution.
So get that problem first. If you're hypotonic, the solution has
Less.
less. So which one, you see B or C, B or D, which one's right for that one?
B.
Now, if the solution contains less, then the cell must contain more. So if you solve it that way, you'd be right every time. If your hypotonic solution, and I did say, I said situation up there, but solution, the water contains less stuff. Therefore, the cell must contain more stuff. So the answer would be Bravo and Charlie.
Bravo what?
Now, if I said hypertonic solution, it'd be right back. More stuff in the solution, therefore, less stuff in the cell. Large particles do what to the rate of diffusion? Hard to move a big old thing around, so the bigger you are, the harder it is to move you, so it's going to slow down diffusion. Most molecules can or can't? Huh?
They cannot.
Y'all saying A or B?
What was that? We need to know
Most of
that.
them cannot.
Yeah.
That's what they have the cell channels for.
Why not?
If most of them could, you wouldn't need to share the cell channels. Facilitated diffusion is still diffusion. So it does not. It's still diffusion. Does not. Those, one's a carrier and one's a channel. Okay, which one's a carrier?
B.
B is a carrier squeezing it through, and a channel is just an open tube. So the answer would be in order: Bravo, and then Alpha. There's some channels that are only good for moving water. What do you call them?
Aquaporins?
Those are aquaporins. Aquaporins. ATP can produce energy, but what else can also? Yeah, hydrogen can also spin a pump, and so can sodium. You know, you ever seen the thing you put in your tire, your bicycle, and the wheel rolling makes electricity, and the little light burns on front, a little generator? Well, anything that can spin that generator, right, will make electricity. Even your hand can do it. Well, ATP can spin it, and hydrogen can spin it, and so can sodium. Iron and calcium don't spin anything. You need both of them. Iron you need in your red blood cells to carry oxygen. Calcium you need in your blood to make it clot, and your nerves to make it carry impulses, and in your bones to make them strong. But in both C and D, it is purely a structural job. But on A and B, those are functional. The function is to make energy. So ATP, but ATP can store it. ATP can save it for a rainy day. The other two cannot. You use them right then. Now those are some choices. And I imagine on my test, I don't have it like this way. I have the question, and these four choices. Question, same four choices. So let's talk about it. All right. When materials are released from the cell, that's the key. Released from the cell. Exo means to exit. Let the words help you now. Don't ignore the words. All right. Evaginates, it sinks in. Materials fall into it or taken into the cell.
Endocytosis.
Huh?
Pino.
You know. All right. Come ahead. All right, this one. Materials are actually attracted. They're sucked in on purpose. They're drawn like a magnet. and then a pit's formed, and you still get to see the magnetized things. Which would that be of those? That's the receptor mediated, because receptor is what controls the formation of the vacuum. I'm right here. Take a large thing in, phagocytosis. Now I got some pictures coming up. What do you see? What do you see happening here? I see a cell spitting something out. So what do you call that? All right. This one, I see the pit forming. I see it pinching off. You're taking in both green, round things and purple triangular things. What would that be? Pinot. I like Pinot. That is just Pinot, I'm sorry. The picture over there is the real thing. Over here, we're trying to show you. Now this one here, there's those attractors. And when it's over, they're still there. I can see them here in the real picture. There is to start to sink in. I still see them right there. I see them, we can draw them pretty. But when they get inside there, see the round nature? It's not full, is it? They're stuck. They're stuck to the attractors. So you're going to see a round look. On the other one, you're going to see it. Hang on, where'd I go? On this, let's see. On this one, it's just full. But on this one, they're always kind of stuck on the edge. Because they are stuck on the edge. And that's why it sounds the ringing. And on this one, there's like an amoeba eating something whole. And that is the phagocytosis. Now, this should be simple using my little Bobby Jeff rule. What's going to happen? It's going to get bigger. It's going to gain water. Now, here's the other half of the one I asked you earlier, but this time it's hypertonic. Work on the solution first, then get the cell second. If you're hypertonic, what's true about the solution? Hyper means what? More. So, what choice is now right? B or D? D is right. Now, if the solution has more, then by converse, the cell must have. So what's your answer now? It'd be A and D. That's why I said, get the solution first. And then flip it. So when you flip it, that's the cell. If solution has more, the cell has less. If the solution has less, the cell has more. If they're equal, then of course they're equal. And that'd be isotonic solution. Well, here I am. What's going to happen isotonic? Nothing's going to happen. It's going to stay like it was. Lower concentrations tend to do what to how fast diffusion occurs? It's going to slow it down. This time I've got 2%, and basically you can say 0%. Distilled water is 0%. There's nothing in it. Now, if you're 0 solution versus 2 the cell, are you hypo or hypertonic? Hypo, because there's less in solution, right? So now, if I put that cell inside that hypo, it's going to be A, B, or C. It's going to be which one? Bravo. It's going to swell up. But you got to first figure out, well, distilled water is 0. The cell is 2. That's a hypotonic solution because there's less depth in the solution, right? More in the cell. Well, if it's hypotonic, Bobby Jeff rule tells you, big O, swell up. There's for the green things like your tomato plants and your black-eyed peas, not the oak trees that you rot leaves off of, but for herbaceous plants, there's water pressure inside. If you stop watering them, they droop over. You water them, they stand back up. But what do you call that pressure?
Sugar.
It's called turgor pressure.
Sugar, whatever.
It's called turgor pressure. The other three words are real,
Plasmolysis.
but they don't apply to the water. Now here we go, 2 and 2, they're the same, aren't they? What do you call that?
ISO.
ISO. So what's going to happen?
Nothing.
Nothing's going to be C.
See.
Nothing's going to happen. Osmo regulation is well, anything osmo refers to what, y'all know?
Water.
Water. Controls water. Those contracted vacuoles inside like Paramecium or Euglena or Volvox, where do those things live? They live in water. So their job is to take the water, get into the cell, and do what to it?
Collect excess water.
Spit it back out. So what's the answer here? They remove excess water. They remove excess water. That's what it's for. You don't have those things in you. Now there is a paramecium. I see the contracted vacuole in both of them. But which one has it full of water?
The one that's swallowed?
This one is full of water. He just got through spitting. So that's the contractile vacuole. He needs a spit. He just finished. Plants ain't got bones. So they use what to hold them up?
Cell wall.
They use cell wall and the central vacuole. Like stacking shoe boxes up. The boxes are the cell wall. And if you had a big stack and they're all empty, they might collapse themselves, right? So if you were to fill them full of shoes, then the shoes might help hold them up. That'd be the central vacuole. So those two things make it. Now, the pine trees out here, and the oak trees, and the ginkgo you brought, they have a different kind of material. It's called woody material. And if you start watering him, he ain't going to flop down, lay on the ground. The problem is, though, in a drought, he'll stand there and be begging. And then when it rains and suck the water up, there's so much water, make the limbs so heavy, they break off falling out. So you're better off having rain all the time than having like 3 months of drought and a hard storm. That's when trees break off. Actually, limbs break off. But a big limb can mess the car up pretty bad, falling 40 feet up. If you don't water your plants, they're going to droop. We call this. But scientists call it what?
Plasmolysis.
Yeah, don't reverse the two words. Commonly, it's called wilting. My grandma once said my plants had they plasmolyzed. She said they wilted. Now, I might tell her scientists I did. All right, got one more to look at.
Plasmolysis.
I made it up. And it's not big. So we have what, 80, and then 35, and then 60. And you know the side of my test, they ain't all on there. All right, let's take a look at this one here. What's B point to?
Nucleus.
I did say structure, but you say nucleus. What's A point to?
Cell membrane.
I'll take cell membrane, or I will take plasma membrane. That's not a cell wall. I didn't tell you what kind of plant that was. If it were an animal plant, never have cell wall. It was an animal plant. That's stupid.
Yeah,
An animal cell never. Plant cell would have cell wall. I just heard myself say that. Which one is cytoplasm? By chance, the letter C is cytoplasm. What kind of cell won't have the nucleus? The prokaryote. You won't have anything. There's no membrane stuff in a prokaryote. What two letters would be incorporated into the term protoplasm?
B and C.
Everything in B and C. A is still not. Cytosol, another word for cytoplasm. Answer C again. Nucleoplasm is found in the nucleus, therefore the answer obviously is going to be Where's the area where you find the organelles of the cell? C. And you know B is an organelle. You know that, right? The nucleus is the cell's biggest organelle. So even it's in C. Cytoplasm. How much water is in it? Between what numbers?
C.
Between, it's actually 80,
It's
right?
about 80, yeah.
Yeah.
So, when I said a little more than, now if I said a little less than, then what you got to put down? I may have changed that something. So when you don't look at the question, oh, I know this one. I may have changed more to less. If you don't read it, you won't see me do that. And as far as color goes, what color is it?
Clear.
Yeah, we stain it to make it. The colors you see, we put dyes. And a dye makes it turn red or blue. But before you dye, it's just clear. You can't see anything.
So if it's colored orange, would all the cells just be like bright orange colors?
Not necessarily. There's some cells that are yellow. Depends on what it is. Cytoplasm does or does not include stuff inside the nucleus? It does not. Cell membrane is more what? You should know about it. It's more fat. All the things are in there, right? There's more fat. All those microfilaments and microtubules, they kind of do for a cell what your bones do for you. What do you call that? A cell, though?
Cytoskeleton?
That's a cytoskeleton. Cyto means cell. You're saying cell skeleton. Cytoplasm contains many salts. Salts make it good or bad? Good.
Excellent.
You need salt. You got to conduct electricity to live. If you don't conduct electricity, like your heart, you're going to die. You're electric. Well, not like electric toaster, but you're electric. Everything between the cell membrane and nuclear membrane would be what? Cytoplasm or cytosol. Either one's fine. If you're inside the cell, including inside the nucleus, then what do you call it? That's protoplasm. You're everything. And the nucleoplasm contains both the chromatin and the nucleolus. But which one turns into chromosomes later? The chromatin does. And which one makes the ribosome? You knew that from previous tests, right? Glucose, too big to enter a mitochondrion, so to make it smaller, it's broken down into, well, what combination? 6 1 carbon, 2 3 carbon, or 3?
Two, three carbon.
2 is broken. First off, glucose is 6 carbons, right?
Yeah.
If I cut it in half, what do I have? 2 3 carboners. And what do you call that? No, those 2 3 carboners, what do you call them?
Isotope?
The pyruvic acid. That can go into the mitochondria. Because you're only 3 carbons big. But that's big old 6 carbon. He's in the cell, but he ain't going any further than that until he's broken in half. And that's called, by the way, glycolysis. And it makes that break. What of those chemicals actually captures and holds energy for you?
ATP.
But they're all real. But ATP captures it. We're going to stop right there. There's no point me going any further than to do that. Y'all going to study now. I'll see you tomorrow.